All right. Hello and welcome back to another episode. So today we're going through the biology of sex determination. So we're going to go into what happens on a molecular basis to define genetic sex, primary sex, and secondary sex. So what actually is the difference between these two, and what are the processes that make them unique entities? All right. So before we go into each one of these individually, and we're going to go into the genetic basis of each of these categories, just want to clarify some definitions first and kind of give some food for thought at the same time. Okay. So genetic sex, okay. Genetic sex is your chromosomal complement at, at birth, right? Or at conception, I should say more correctly. So for most individuals, XY for a male and XX for a female. Okay. Of course, there are variations of this, such as in Turner syndrome, you get X naught, right? Only one X chromosome. Or in Klinefelter, you'll get XXY. Okay. But for the majority of people, XY defines a male and XX defines a female. Again, we're going to go into so much more detail in a bit. But yeah, essentially, genetic sex is your chromosomal complement at birth. Okay. Your primary sex. Okay. These are, this is categorized by your genitalia at birth. Yeah. So typically, males will develop with testes, whilst females will develop with ovaries. Okay. Again, speaking in majority of cases here. All right. And then finally, we have secondary sex, right? Which is your hormonal changes, right? Hormonal changes at puberty. So now it may seem at first that, well, these three are all interconnected, right? That you would think, all right, so if someone has XY chromosomes, right, they're male, so at birth they should have testes, and at puberty they should have their voice drop and their muscle bulk increase, right? But interestingly enough, what we're going to go through today is how each of them are distinct processes, right? And there are certain conditions where you can get the genetic sex of a male and the primary sex of a female, for example. Yeah. Or vice versa. Or you can have a primary sex male who develops female characteristics at puberty, right? That's what we're going to go into today. What is the genetic development of those conditions? We're not going to go into the specifics of those conditions, but rather what are the changes at each of these steps? And that kind of sets the foundation of what can go wrong. All right. So in more detail on genetic sex. Okay. Genetic sex. Now, we said, of course, that we said earlier that males typically are XY, females are XX. But then it begs the question: why is I gave the example of Turner syndrome, right, and a condition of aneuploidy. Why would X naught be considered female? Why? Versus Klinefelter being XXY. Why is that male? Right. So the widely accepted definition is just the presence of the Y chromosome. Okay. The presence of the Y chromosome equals male genetic sex. Yeah. So typically, if you had some rare genetic conditions where you had like 5X chromosomes, right? 4X chromosomes, which is actually possible. Very, very incredibly rare, but possible. You would be considered female because you lack the Y chromosome. Whereas if you had 4X chromosomes and a Y chromosome, you would be considered male because of the presence of a Y chromosome. Genetic sex is thankfully probably the simplest because of that. It's as simple as presence of the Y chromosome versus its absence. All right. Where it starts to get more difficult is at primary sex. Okay. Primary sex. And we said just quickly, we said that was the development of the genitalia at birth, the development of the gonad. Okay. So, what is going to drive male gonads versus female? Well, if we've said that we define male genetically as XY, hopefully it should make logical sense that there's something about the Y chromosome that is driving male development. And that's actually true. Okay. So primary sex. So on the Y chromosome, we have a region, okay? This region, which we denote as SRY, the sex determining region Y, okay, has that gene, the sex determining region Y gene. It's only one exon long. And when expressed, it is critical, okay, critical for testes development. Okay. Why, you may ask? What does it actually do that's so critical for testes development? Well, it does two things, right? Well, it may do more, may do less, but the consensus is that there are two known mechanisms, okay? Right? The first is it's a transcription factor. And it has effects on Sertoli cells and AMH expression. Okay. So it's critical, right, for the development of Sertoli cells, and it is a transcription factor that is necessary for anti-Müllerian hormone expression. And we're going to go into anti-Müllerian hormone in a bit more detail in a second. All right, so we have this sex-determining region Y, and we know it's critical for the development of the testes because it encodes for the activation of anti-Müllerian hormone and Sertoli cells. Okay, so when it comes to development of the gonad, right? Obviously, the first thing that needs to happen is we need to have our germ cells migrate, yeah, towards the gonad. To gonad. Now, in a male, okay, with high expression of sex-determining region Y, yeah, we're going to typically have testes development. Whereas in a female, in the absence of SRY, we get an absence of testes development and we get ovarian development. Okay, now there's another key gene here called DAX1, which is high in females and low in males. Okay, so the DAX1 gene, we say, inhibits the development of testes. So when we're trying to develop testes to form a male, we want high expression of the sex-determining region Y, and we want low expression of DAX1. In other words, we want high of the gene that promotes testes development and low amounts of the gene, DAX1, that inhibits it. Okay. And so as we said, this causes the growth of testes. For a female, it causes the growth of the ovary. Now, the astute of you guys may be thinking, well, just because you have an inhibition of testes, why would that mean ovaries then grow? We're not really, from this specified mechanism, we're not really driving the growth of something new. We're just stopping the growth of an existing structure. So how are ovaries growing? It's actually not yet known. So the genetic induction of the ovary is actually poorly understood. At a simple level, this is the basis of what is widely accepted at this moment in time. Yeah, in the absence of these key genes, the ovaries will develop. Okay. We'll go into the female now in a bit more detail because it's a bit simpler than the male. And then we'll go into the harder processes of the male. Okay, so like I said, we've just expressed key genes / not expressed others to produce the ovary. Okay, now what does the ovary do? It produces, the ovary has its key cells, first of all. We know we have theca cells and we have granulosa follicular cells. Granulosa. Okay. Now, each of these cells are responsible for steroidogenesis. Really quickly, not the topic of this video, but we know the theca cells can make like testosterone, but in the lack of aromatase activity, they can't make estradiol, gets transferred to the granulosa cells, which like other enzymes that can't actually get to the progesterone precursors, but they do have the aromatase, so they can take the testosterone from the theca cells and convert them into estrogen. All right, so the net effect is just estrogen production, okay? Once we have high levels of estrogen, we have a number of processes that start to form, okay? One, we have the Müllerian duct. Müllerian duct becomes stimulated. And this is then going to cause a cascade of events that cause differentiation into structures of the female reproductive tract. We're going to go into that in a second. And of course, the opposite, we need to cause a regression of the Wolffian duct. Yeah. And then we have the urogenital sinus, which is going to form part of the labia and the lower part of the vagina and the clitoris as well. Okay, urogenital sinus, that is. All right, so just into more detail on the Müllerian duct for a second. What is the Müllerian duct going to form? Okay, well, the Müllerian duct is going to form your upper vagina, your fallopian tubes, and of course your uterus. Okay. And we said, noting again, just quickly, we said the lower vagina is going to come from the urogenital sinus. Okay, so how has this happened, right? We said low SRY, high DAX1, we've formed our ovaries. Our ovaries have our theca and our granulosa cells. They produce estrogen. And then we have regression of one duct and differentiation of the other. So in a female, noting that the Wolffian duct regresses, it atrophies, it undergoes apoptosis, goes away, whereas the Müllerian duct stays a feature within the female, okay? And it's really important to understand this because in a male, it's the opposite. In a male, the Müllerian duct is going to regress and the Wolffian duct will remain patent. All right, so on that note, let's talk about the male. All right, so we've developed our testes, right? We had at high SRY, we had low DAX1. Okay. Now our testes have two main cell types as well. We have our Sertoli cells, and we have our Leydig cells. Our Leydig cells also, you may see referred to as the interstitial cells of Leydig. Okay. So now the Leydig cells, so before we go into that, the Leydig cells, we know out of the context of genetic development, they're in charge of producing testosterone, whereas the Sertoli cells are responsible for spermatogenesis, okay? So the Leydig cells, what happens? We have testosterone being produced. Okay. And we actually have a little bioreaction going on too, right? Testosterone in the presence of 5-alpha reductase, an enzyme, will form dihydrotestosterone, okay, which is about 30 to 50 times more potent than testosterone itself, okay? Now, so what do these actually do, okay? Well, testosterone causes the differentiation of the Wolffian duct. And what do these actually form? Well, the Wolffian duct is going to go on to form the internal structures of the male reproductive tract. Okay, so your ductus deferens, epididymis, and your seminal gland, also known as your seminal vesicle. All right. So what does dihydrotestosterone do? Well, that produces your external genitalia. So the dihydrotestosterone is going to go on to make the penis and the scrotum. Awesome. Now, the Sertoli cells are a little bit more complicated. The genes that are involved require specific sequences, right? It requires a more careful control, I'll say, of DNA expression to go on to produce proper primary sex development, i.e. proper development of the male reproductive tract at birth, okay? So we have key genes that I'll put in a different color here, okay? WT1, okay? SF1 and SOX9. Okay, so WT1 and the SF1 genes must be active together, and by doing so, right, by being expressed together, they're able to activate SOX9, the SOX9 gene, okay? And now this SOX9 gene is then able to cause an activation of AMH expression, okay? So this causes an activation of anti-Müllerian hormone. And hopefully this rings a bell. We spoke about the Müllerian duct being specialized in a female to produce the structures of the female reproductive tract. Whereas in the male, we don't want those structures. We want the Müllerian duct to regress. So we produce anti-Müllerian hormone. Hopefully this makes sense, okay? And that causes regression of the Müllerian duct. Awesome. And so now we should have our primary sex characteristics done, okay? So just a quick summary of where we are so far. The genetic sex, right? Chromosomal complement, presence of a Y chromosome. We said they were male. Primary sex, so what reproductive tract were they born with at birth, okay? And in the testes, to grow the testes, we needed low DAX1 and we needed high SRY, okay? And then once we form those testes, we have the testosterone producing our internal structures, the dihydrotestosterone producing the external structures, and these key genes, WT1, SF1, SOX9, causing anti-Müllerian hormone to regress the Müllerian duct. In a female, it was the opposite. High DAX1, low SRY expression caused development of the ovaries, and we wanted to keep the Müllerian duct to make the structures of the internal tract, okay? and we wanted to regress the Wolffian duct, so we didn't grow the male reproductive tract. And then, of course, we had elements of the urogenital sinus producing other structures, of course, such as the labia. Perfect. So that's our primary sex. And thankfully, the last one, secondary sex, is more simple. This is the hormonal changes that occur at puberty. So it's simply to do with hormonal signaling or changes. Okay. So if we have testosterone at puberty, right, the typical features of a male at puberty will occur, right? Their voice will deepen, they will grow in muscle mass, they will change to a more catabolic state, increasing their enzyme activity of things like UDP glucuronidase or glucuronidase or anything like that. Whereas in a female, if we have estrogen, we're going to cause like change in fat distribution, right? We're going to lower the cholesterol, widen the pelvic inlet and outlet, right? We're going to cause like vaginal lubrication, things like that, drive breast development, typical changes that occur at puberty. All right, and that's the genetic basis of the sexes. And yeah, like I said, we're not going to go into it here, but if you understand each of these three processes, you know where things can go wrong. And that's the basis of a number of the genetic diseases. All right. I hope that was helpful. And yeah, stay tuned for the next video.